Welcome back to G Fun Facts Online. Today, we are going deep into a subject that affects millions of people, yet remains one of the most misunderstood and poorly treated conditions in modern medicine. We are talking about chronic pain, but we are not just talking about the problem. We are talking about a revolutionary solution emerging from the labs of Sydney and bioprinting facilities around the world. We are talking about the dawn of the cellular pain sponge. To understand why this is such a massive paradigm shift, we first need to strip away the common misconceptions about what pain actually is. In a healthy body, pain is a gift. It is an essential, sophisticated alarm system. Imagine you touch a hot stove. Before your conscious brain even registers the heat, a reflex arc in your spinal cord has already commanded your muscles to yank your hand away. This is called nociceptive pain. It is functional, it is protective, and most importantly, it is temporary. Once the tissue heals, the alarm stops ringing. But for millions of people, this alarm system breaks. This is what we call neuropathic pain. It is not a symptom of an injury that is currently happening. It is a disease of the alarm system itself. Whether it is caused by diabetes, chemotherapy, a traumatic spinal injury, or a viral infection like shingles, the mechanism is roughly the same. The peripheral nerves, the wires carrying signals from your body to your brain, get damaged. And in a cruel twist of biology, instead of going silent, they go rogue. They start firing spontaneously. They send a constant, relentless barrage of danger signals to the spinal cord, even when there is no danger present. Now, here is the detail that usually gets missed, and it is the key to understanding the new cure. Normally, your spinal cord acts as a gatekeeper. It is not just a passive cable, it is a processing center. Inside the spinal cord, you have a specific type of cell called inhibitory interneurons. Their job is to say no. They release a neurotransmitter called gamma-aminobutyric acid, or GABA. Think of GABA as a chemical brake fluid. When a minor signal comes in, like the gentle brush of a cotton shirt against your skin, these GABAergic neurons fire. They apply the brake. They dampen the signal so that it doesn't travel up to the brain, to be perceived as pain. But in chronic neuropathic pain, this braking system fails completely. The constant bombardment from those damaged rogue nerves causes these precious GABAergic neurons to die off or dysfunction. The gate is left wide open. The chemical brake is gone. This leads to a condition called allodynia, where things that should be painless, like a breeze or a bedsheet, feel like sandpaper or fire. The system has lost its ability to self-regulate. For the last few decades, our medical response to this precise, localized circuit failure has been incredibly crude. We have been using a systemic sledgehammer. We flood the entire body and brain with opioids. Opioids work by binding to receptors throughout the entire central nervous system, dimming the lights in every single room of the house, just to fix a flickering light bulb in the hallway. The result is addiction, tolerance, respiratory depression, and a brain fog that steals the patient's quality of life. The unmet need here is staggering. We have severed nerves, we have implanted electrodes, and we have prescribed endless cocktails of drugs, but the pain remains. This brings us to the revolution. A team of researchers leading the charge from the University of Sydney asked a fundamental question. Instead of drugging the brain to ignore the signal, why not rebuild the brake? This is the concept of the cellular pain sponge. The idea relies on a convergence of stem cell biology and advanced tissue engineering. The goal was audacious: to take a patient's own cells, perhaps from a skin biopsy or a blood draw, and transform them into the specific type of neuron that has died off, the GABAergic cortical interneuron. If we could manufacture these peacekeeper cells and transplant them back into the damaged spinal cord, could we restore the lost inhibition? The answer, according to landmark studies, is a resounding yes. But the process of getting there is essentially biological alchemy. It starts with induced pluripotent stem cells, or iPSCs. This technology, which won Shinya Yamanaka a Nobel Prize, allows scientists to take a mature adult cell and rewind its biological clock. They reprogram it back into an embryonic-like state. This solves two massive problems instantly. First, it bypasses the ethical issues of using embryonic tissue. Second, because these cells come from the patient's own body, the genetic match is perfect. There is no risk of immune rejection. But having a stem cell is just the beginning. A stem cell is like a student entering kindergarten. It has the potential to become anything: a heart muscle, a tooth, an eyeball, or a neuron. The researchers had to figure out the exact curriculum to turn that stem cell into a pain-killing specialist. They developed a protocol to bathe these iPSCs in a specific cocktail of growth factors and chemical signals. Day by day, over weeks, these cells are nudged toward the neural lineage. Then they are pushed to identify as spinal cord cells. Finally, they are locked into the phenotype of a GABAergic interneuron. When tested in the lab, these bioengineered cells did exactly what their natural counterparts do. They acted as biological pumps, releasing GABA, the body's natural painkiller. But the real test was in the living organism. When these cells were transplanted into the spinal cords of mice with severe nerve injury, the results were nothing short of miraculous. This is where the metaphor of the sponge comes to life. The transplanted cells didn't just sit there, they integrated. They grew dendrites and axons, weaving themselves into the host's damaged neural circuitry. They effectively soaked up the excess excitatory signals. When the damaged nerve screamed pain, the new GABAergic cells released their calming neurotransmitters, neutralizing the signal before it could ascend to the brain. The mice, which had previously flinched at the slightest touch, returned to normal behavior. The pain was not masked, it was not dulled, it was resolved. And critically, this relief was lasting. A single treatment provided permanent resolution of symptoms in the animal models. There were no side effects. The mice didn't get high, they didn't lose motor function, and they didn't develop tolerance. The sponge only worked when there was a spill of pain signals to absorb. It is an intelligent, on-demand therapy. However, biology is only half the battle. You cannot simply inject a slurry of neurons into a human spinal cord and hope for the best. The spinal cord after an injury is a hostile environment. There is inflammation, scar tissue, known as glial scarring, and a constant flow of cerebrospinal fluid that can wash loose cells away. To make this a clinical reality, we need a structure, we need a home for the cure. This brings us to the second pillar of this technology, the analgesic scaffold. This is the realm of bioscaffolding and material science. Modern analgesic scaffolds are marvels of engineering. They are typically hydrogels, networks of cross-linked polymers that mimic the soft, squishy texture of the central nervous system. These scaffolds are often 3D printed or cast with microscopic precision. They are porous, like a coral reef, providing tiny nooks and crannies for the stem cells to nestle into. But these scaffolds are not just passive houses. They are functionalized. They are impregnated with neurotrophic factors, which are essentially biological fertilizers that encourage the stem cells to survive and grow. Some of these scaffolds are even electroconductive. Since neurons communicate via electricity, a scaffold that can conduct a charge helps the new cells synchronize with the host's existing electrical rhythm. In cases of severe spinal cord injury, where there might be a physical gap or cavity in the cord, the scaffold plays a structural role. It acts as a physical bridge. Imagine a 3D printer laying down a lattice of biodegradable polymer. In one ink cartridge, there is the structural gel. In the other, the living GABAergic neurons. The printer constructs a living patch, layer by layer. This patch is then surgically implanted into the site of injury. Over time, the scaffold is designed to degrade. It dissolves at the exact rate that the new tissue forms. By the time the polymer is gone, the new neurons have established their own matrix. The scaffolding is removed, but the building, the restored inhibitory circuit, remains. The social implications of this are profound. We are currently living through the opioid epidemic, a crisis fueled by our lack of better options. Opioids are a blunt instrument. They dampen pain perception, but they cause opioid-induced hyperalgesia, a condition where the drugs actually make the patient more sensitive to pain over time. The Cellular Pain Sponge offers a restorative therapy. Because GABA is the body's natural inhibitor, it does not activate the dopamine reward pathways. There is no high. There is no addiction. Because the cells are living entities, they regulate their own production of GABA based on demand. They do not wear out like a drug receptor, so there is no tolerance. And because the cells are placed specifically in the dorsal horn of the spinal cord, the processing center for pain, they do not affect the rest of the body. No constipation, no respiratory depression, no brain fog. For a patient with neuropathic pain from a spinal cord injury, who might be facing 40 years of daily agony, this is the difference between a life managed by sedation and a life fully lived. So why can't we book an appointment for this tomorrow? We are currently in the valley of death, the translational gap between a successful mouse model and a human therapy. The primary hurdle is safety. Pluripotent stem cells have the capacity to divide indefinitely. If a single cell fails to differentiate fully into a neuron and remains in its stem state, it could theoretically form a teratoma or a tumor. The researchers have spent years refining purification processes, using magnetic sorting and chemical kill switches to ensure that only the finished, non-dividing neurons make it into the scaffold. Studies in rodents and pigs have shown no tumor formation, which is a critical green light. We also need to prove durability. We need these cells to last for decades in a human, not just months in a mouse. And finally, there is the cost. Customizing cells for every patient is slow and expensive. The future likely lies in off-the-shelf allogeneic cells, universal donor cells that have been gene-edited to be invisible to the immune system. We are standing on the precipice of a new era. For thousands of years, we were chemists, mixing potions to trick the mind. Now, we are becoming architects. The cellular pain sponge represents a shift from pharmacology to biology. We are no longer trying to suppress the symptom. We are repairing the machine. Imagine a future where a diagnosis of diabetic neuropathy or a spinal cord injury does not carry a life sentence of chronic pain. Imagine a soldier surviving in blast, not to return home to a haze of oxycodone, but to receive a bioprinted scaffold that knits their nervous system back together, silencing the phantom fire before it can burn. The science is real. The cells are ready. The scaffold is printed. The silence is coming. Thanks for listening to G Fun Facts Online.